Well, thank you all very much for joining us today on this webinar. As you can see, it's about the psychophysiology of emotions. So to also see this connection between our emotions and our body, so to say. I will be your host today, so I will guide you a bit through this webinar, but the presenter today is Patrick Zimmerman. Patrick already has 16 years of experience at Noldus as a consultant. So he is indeed the right man for the job to tell you how this all works. Let me just give you a short idea on the rules today that we have, to give it a bit more structure and to guide everyone through this webinar in an easy way. Okay, so as Patrick will guide you through the story itself, we will introduce some polls along the way. So we'll ask some questions from you, have some feedback. So that's going to be nice because, we always try to keep it interactive, using the latest technology like everyone is doing. We see them a lot in the education as well. I would like to ask you to put your questions in the Q&A. You can find a button for that below. These questions, we will try to answer as much of them as we can at the end of the webinar. In case there might be a lot of questions, we can also answer them one-on-one. You can talk to us outside of this session. But feel free to post any questions because they might be very relevant also for the other participants. So feel free to put anything in there. All right, then that is it for my part by now, and I would like to hand it over to Patrick.
Okay, thank you very much, Guy. Thanks everyone for joining this webinar on the psychophysiology of emotions. Let's get started. Well, I'd like to point out that no children were harmed while preparing this webinar. In this webinar, I will talk about the possible relationship between physiological measures and emotional states. For example, does sadness, as you can see on the face of this kid who was subjected to a harmless version of a lie detector for a children's program, does this sadness show up physiologically with a kind of specific dynamic pattern? And those of you who attended a previous webinar on how to measure emotions might recognize some of the slides I will show you, because the current webinar is very much linked to the previous one on emotions. So this is the outline of the webinar. I will present for about 30-35 minutes. And after that, there is ample time to answer your questions that you, as Kai pointed out, can put in the Q&A. I will start with telling a bit about our company, Noldus IT, and a bit about myself. Then we go quite a bit back in time to see how evolution has shaped our brains and the way we respond to the world around us. And then we will move on to psychophysiology, what it is and how it might be related to specific emotional states. And I will conclude this session with some conclusions and recommendations. For those of you who are not familiar with our company, we are an IT company, Noldus Information Technology, and we provide tools and services for behavioral research, both in the animal and human domain. And our headquarters are based in Wageningen, the Netherlands. It's a small university town on the east side of the Netherlands. And we also have offices in China and the US. We were founded by our CEO, Lucas Noldus, in 1989. And currently we have about 160 people working at Noldus IT from 9 different countries. And we also have a large customer base in 98 countries all over the world. So the countries in which we have customers are highlighted in green in this world map. As Kai already mentioned, I've been with Noldus for over 16 years now. I was trained as a behavioral biologist, and I currently work as a trainer and consultant at Noldus IT. So working for Noldus has taught me a lot about human behavior and psychology as well, because I've been trained as a behavioral biologist for animal behavior. And I've kind of developed a particular interest in why humans behave the way they do, both from a professional but also a personal point of view. So let's look at the evolution of the human brain to start our journey. Natural selection has shaped our brain with structures and functions that benefit our survival. So a million years of evolution has resulted in a human brain that consists of kind of three basic parts. The oldest part, the part in a brown color, is what we call the reptilian brain, and which is responsible for a species typical instinctual behaviors. A newer part is the part shown in red in this picture, is what is called the limbic system, which is responsible for motivational and emotional aspects of human behavior. And the latest addition to the mammalian brain is the neocortex, which is only found in higher animals and is responsible for rational thinking. And these parts are anatomically not as distinct as shown in this kind of schematic picture, but are more integrated also functionally. Another more recent conceptual view of our brain was put forward by Daniel Kahneman, a behavioral economist. And he makes a distinction between system one, which is represented in the previous slide by the older parts of the brain, so the reptilian brain and the limbic system, which responds in an instinctive, subconscious way, without involving much deliberation or thinking. And on the other side, there is system two, which is the thinking part of the brain, if you will, which is represented by the neocortex. This part of the brain is slower and takes more effort to be activated. So from an evolutionary point of view, it's more efficient and adaptive to be governed by system one, the more subconscious, instinctive part of our brain. As a result, humans generally respond in an irrational, emotional way to what happens around them. And we humans tend to create the story afterwards using system two to make sense of our behavior and also justify it. On a more physical or physiological level, our instinctive natural responses to anything we encounter in our surroundings are shaped by our brain and can be categorized in a fight, flight, or a freeze response. And these responses were shaped by evolution and helped us deal with threats and dangers and helped us to survive. And early in human evolution, these threats were real threats, such as predators, so animals that want to eat us as a food item. But I think it's also good to point out that nowadays these threats can be have a different kind of nature. So these could be an aggressive driver on the highway, or another threat could be the thought of this annoying colleague that you encounter every day in the office. So let's look how our natural responses are adaptive. So if we look at the freeze response, freezing gives you time to assess the situation or makes you less conspicuous or detectable to a predator. So here you can see a freezing response of an animal. A lot of animals show this freezing response when they are threatened by a predator. When we look at kind of a human freezing response, I think we all know the response we get when you certainly get a fright because of a loud noise or someone jumps on you from behind. You just kind of feel your body freezing. In an extreme case, our autonomic nervous system, which I will come to later in this presentation, can shut down completely, and which can result in you fainting, so really collapsing to the floor. In a less extreme situation, we can see, for example, someone freeze in her chair, so really holding on to the chair in response to something that's not very pleasant. the flight response is adaptive in getting away from a threat and not getting killed. Like in this picture, someone getting away from a bull chasing him. But we can also show more a subtle flight response where we do not actually get away from a threat, because that would not be socially acceptable in this case. But we show subtle signs that we would like to get away from the other person. And finally, the fight response was initially necessary to confront a threat or a danger. But this does not always involve physical interaction, but can also involve words, so language. So in terms of a fight response, in terms of body language, arguing or verbal fights can be an example of a fight response. So please keep in mind that although these natural responses evolve to cope with physical threats and dangers, these natural responses also occur to mental threats. And I think that's more likely to happen in current society, in current conditions we live in. Threatening, unwelcome thoughts evoke the same natural response as an evolutionary older threat. So let's move on to psychophysiology. So physiology concerns the study of bodily functions and how they work. So they look at neurotransmitters, physiological phenomena such as functioning of the heart, respiration, but also the hormones. And psychophysiology ties this physiology to psychological phenomena. So how does a person emotionally respond to stimuli in her environment, and how does this affect her physiology, and also vice versa? So really, it's about the relationship between mental processes and physiological phenomena. So Kai, I think it's time to start the first poll. And this is about, what type of physiological measures do you measure in your studies, if any?
Of course, Patrick. It's about the different measurement devices. So I will launch this poll and you can enter which of these devices that also you use yourself. So we can give it a few minutes. So there's a lot of answers coming in. Thank you. Right, maybe a few people are still a bit shy to give an answer, but we have absolutely no idea who put in what, so don't feel pressured. And you can also put in multiple answers. They are possible. All right, so I think we have most people. Just a few. Okay. So Patrick, you have any thoughts on what you've already seen so far?
How do I see the results of the,
okay.
poll?
I thought they were also visible to you, but maybe they're
No.
only to me. Okay, I will end in a minute, and I will probably provide you a bit on what the answers are. So we see that a lot of people, 55% even, they are already using electrodermal activity and galvanic skin conductance. So It's quite a high percentage. Facial EMG, also more than 50%, more than 50% on heart rate pulse. And, you know, slightly below the 50%, also see ECG, and respiration, 42%. So there is quite a lot of use already of these different devices. Like most of these devices, they already are scored around 50%.
Okay.
So I will end the poll now because I think that the people who haven't answered yet, probably they don't use any of these devices as of this moment. But it's interesting to see these different figures. As I end them now, I think I can share these results with you.
Okay. Yep.
So they should be visible. Always interesting to see, you know, what's happening also with your colleagues, right?
Yep. Okay, that's good because I have some more. Well, some people already work with it, so I will provide some more information on heart rate variability, and EDA. So for some people, it might be familiar. For some, it might not be. So let's continue with the presentation. There will be another poll later in the presentation. Well, the most commonly used measures to quantify the activity of the autonomic nervous system, because that's, you know, the system that's responsible for the fight, flight, and freeze response. The commonly used measures are heart rate and heart rate variability, but also electrodermal activity, also known as galvanic skin response, galvanic skin conductance. So I will provide a bit more background information on the autonomic nervous system and these measures: heart rate variability, and electrodermal activity. Well, as the main aim of our brain is, you know, to keep us alive through our natural responses, these natural responses start with a reaction of the autonomic nervous system, which helps our body to prepare for the appropriate action under different circumstances. So we can see two parts of the autonomic nervous system. On the left side, you can see of this picture, you can see the sympathetic part of the autonomic nervous system. This helps you to prepare for your body for action. So typical reactions of this part or this branch of the autonomic nervous system are pupil dilation, increase in heart rate, increase in breathing rate. So it really prepares your body for action. And therefore, the sympathetic branch is kind of, we could say, responsible for the fight-flight response. In contrast, the other branch, the parasympathetic part of the autonomic nervous system, here displayed on the right side of the picture, is involved in the freezing response and a rest digest response. So heart rate slows down when this parasympathetic branch gets activated, and the digestive system gets more energy. So activation of the parasympathetic branch of the autonomic nervous system helps us to recover from, an active period or bodily action. So in general, our body aims for a balance or homeostasis. So it tries to keep the balance between the two branches of the autonomic nervous system, but at the appropriate moment, one of the branches is ready to kind of be activated, to kick in. And when we look at heart rate, the heart as a kind of a big muscle, and heart rate is controlled by two branches of the autonomic nervous system through different neurotransmitters. And you can see a schematic picture on the slide. So the sympathetic branch works through the release of norepinephrine, which increases heart rate. Activation of the parasympathetic branch through the neurotransmitter acetylcholine decreases heart rate. So in a resting state, both branches are equally active in balance, which results in a heart rate oscillating around its resting value because of the equal influence of both branches of the autonomic nervous system. And therefore, the variability of the resting heart rate is usually high. So this is an example of how you can measure heart rate using a PPG sensor or a photoplethysmogram sensor. It measures basically the kind of the flow of blood through a finger, in this case, with a finger sensor. And typically you use equipment with a sample rate of between 250 and 500 hertz. Another, you could say, more accurate way of measuring heart rate, because it measures the electrical activity of the heart. ECG stands for electrocardiogram. This is a type of measurement that you can use to not only measure heart rate and heart rate variability, but also to monitor the functioning of the heart. And this is applied in a medical context. Because heart rate can be very precisely measured with the electrocardiogram, the ECG. ECG is suitable to also quantify the heart rate variability. An equipment that you can use to measure ECG, to measure heart rate using ECG, typically uses sample rates between 500 and 2000 hertz. So it has a higher sample rate because it requires a more accurate sampling of the activity of the heart. So heart rate measured through ECG is based on detection of the R peaks. So here you can see the kind of heart wave with different elements. And the R peak is part of the QRS wave. The QRS wave represents the ventricular depolarization, so depolarization of a part of the heart muscle, which is followed by ventricular contraction. And this contraction results in oxygen-rich blood being pumped from the heart into the rest of the body. So you can see here consecutive heartbeats represented by the R peaks. So this is the way that the ECG algorithm is able to quantify heart rate and heart rate variability. So the heart period, as it's called, or interbeat interval, is the interval, the time in milliseconds between adjacent heartbeats, measured on the basis of the R peaks. From this heart period, the heart rate is derived. So a change in heart rate can be caused by a change in, sympathetic activity, a change in parasympathetic activity, or a change in both. Heart rate variability is the beat-to-beat variation in the time between consecutive RR intervals. And for example, this heart rate variability can be quantified or measured by, here are two examples of time domain measures, the SDNN or the RMSSD. So this is just a way to quantify the heart rate variability. So heart rate, increased sympathetic activity seems to be an indicator of mental stress, and increased sympathetic activity is reflected in a lower heart rate variability. So you can see here in this graph, this is the active period where the heart rate variability expressed as the RMSSD, one of the time domain measures, is relatively low compared to the resting period. So that's highlighted with the blue line here. So the recovery and resting period is characterized by a higher heart rate variability. And what is also important to point out is that the way our higher brain regions, our neocortex, interprets the physiological changes that we kind of experience partly determines the perceived stressfulness of a situation. And one example is a study that was done in the 60s where participants were injected with epinephrine or adrenaline and then put in different contexts. So one group of participants was kind of put together at a table with an angry person, and the other group were put at a table with a happy person. And depending on the situation, the participants reported to feel more angry when they were with the angry people, in contrast with the people who were put together with a happy person. They reported or they interpreted their kind of aroused feeling with a feeling of being more happy. And although both groups were treated with the same substance, they both reported different feelings or different emotional states. So let's move on to EDA, or electrodermal activity. So the electrodermal activity measures the activity of the eccrine sweat glands, which have the highest density on the hands and our feet. And they predominantly have a sympathetic innervation. So there's a high correlation between sympathetic nerve activity and EDA. So you can see a typical raw tonic slow EDA graph here. So these are two different graphs for different participants. And what we see is on this slow tonic graph, we see more fast phasic changes that are often the result of specific stimuli being presented to a participant, to a person. And typically, EDA is measured using two electrodes that are usually put on the hand, either on the fingers or on the palm of the hands. And these electrodes quantify the changes in the conductance between them. And this way you can quantify the activity of the sweat glands as a result of a change in sympathetic nerve activity. And what is an important feature of EDA or galvanic skin response? It's very sensitive, but very non-specific. So if you expose people to certain kind of questions, have you ever cheated on a test, or how do you feel about your mother, or you pinch them, you will see kind of similar responses of the EDA graphs. So the EDA will respond to certain stimuli that kind of arouse or activate the sympathetic nerve, part of the autonomic nervous system, but it will do that based on different types of stimuli and show the similar response. So it's very important that you design your experiment carefully to kind of distinguish different types of stimuli. Okay, before I move on, time for the second poll.
So I will launch that one as well.
Okay, thank you.
And there we go. So thank you so much for the response in the last poll. Let's see what the voting brings up now. All right, already quite some responses coming in. A few people still need some time. So thank you so much for your answers. If those people who did not yet find a good response, you could also please fill in the poll. All right. Giving it the last few seconds for those who still want to provide an answer. And else we assume that maybe the answer that you would want to give is not in there, but there can always be some other questions in the Q&A, of course, that you want to ask. So I will close the poll for now. Thanks everybody for answering. So Patrick, it also helps to understand, what are people working on and working with in the field of studying human behavior. And as I share the results, it's interesting for all of us to see, okay, what are the main types of measurements currently used within the group we have today?
Okay. So, well, it's interesting to see, and that's also what I will talk about in the remainder of the talk, the combination of behavior and physiology. That's something that, we see increasingly, that people combine behavior and physiology, maybe also self-reports. Well, cool. That's interesting. Some people just study behavior. Maybe this presentation will kind of stimulate them to maybe also think about physiology, but let's see. Okay, I'll proceed with the kind of the last part of this presentation. So please put your questions or comments in the Q&A so we can try to answer them or respond to them after I finish my presentation. So in the last part, I will try to kind of link the psychophysiology with kind of measures of emotions or emotional states. And as we have seen in the previous webinar on emotions that I already mentioned at the beginning, the classical view on emotion states some stereotypical facial expressions related to the basic emotions as proposed by Ekman and co-workers. But research in the past decades have shown that there is a lot of variation of how humans express their emotional states. So I think we all know from our own experience that anger, for example, can be displayed in different ways, or happiness. One person could just smile or shout from happiness. Another person starts crying tears of joy. And the same applies to anger, depending on the context. A person might express her anger in different ways. When it comes to physiological responses to emotional states or related to emotional states, the story is slightly different. And as we've seen, there are basically three natural responses: fight, flight, or freeze. So there are not so many flavors in this respect. So your body can be kind of prepared for action or can be inactivated. Your heart rate can go up or down. EDA or skin conductance can increase or decrease. But obviously, researchers have tried to search for certain psychophysiological patterns in relation to specific emotional states. So is anger displayed, or is anger expressed in a specific dynamic change in heart rate, skin conductance, respiration? In their review paper from 2019, Lisa Feldman-Barrett and co-workers looked at studies trying to assess emotions based on dynamic changes in the autonomic nervous system. And maybe a bit disappointing, but on the other hand, maybe also not completely unexpected. Meta-analysis of hundreds of experiments showed that currently there is no reliable relationship between an emotion category such as anger and a specific set of physical changes in the autonomic nervous system. Looking at these studies that have been done, in anger, in an angry emotional state, for example, skin conductance can go up in one study, go down in another, or stay the same in a third study. So this means that changes in skin conductance are not consistently associated with anger. And a rise in skin conductance is not unique to instances of anger. It can also occur during a range of other emotional episodes. So changes in skin conductance are not kind of specific for anger, but can occur in different emotional states. And something similar applies to heart rate and respiration. So they conclude, and it's also kind of supported by other studies, that there is, well, it's very difficult to find a specific relation between a specific emotional state and a dynamic, kind of specific dynamic change in physiological measures. What we've seen and what people find is that sometimes within a study, heart rate and EDA patterns might distinguish emotional categories or are specific for in a response to a specific stimulus or product. But it's often that these results are not replicable across studies, and instead vary across studies, even when studies use the same types of methods and stimuli and use a sample from the same population of participants. So there's a lot of variation between studies and between participants. And it seems that this variation in autonomic nervous responses depend on not only the subject, but also the context, on the, what type of lab you use, how do you deal, how you treat your participants. So maybe this is a bit of a disappointing conclusion, but it's just the reality we have to kind of face, that there is no relation or kind of no general relation between a specific emotional state and physiological changes. So, well, that's the kind of the main conclusion. There is no one-on-one relationship between physiological changes in emotional categories. So does that mean that it doesn't make sense to look at the relation between behavior, emotional states, and physiology? Well, I don't think it doesn't make. I don't think it. I do think it makes sense because it's still useful to look for changes in physiological and or behavioral measures within a participant and a study, to draw conclusions within a study, and to try to search for differences in these behavioral and physiological responses between different stimuli or different products you present in a specific study. So it's still worth looking at these relationships for your specific in your specific study or experiment, and therefore we kind of propose or advocate a multimodal approach. So physiological measures might reveal responses which are not displayed behaviorally. So I think people who study behavior, and I guess that's most of you, all know and have experienced that sometimes you don't see an overt response in your participants. Whereas, when you measure heart rate or skin conductance, you do see a response, and maybe also a different response to different stimuli on the inside. And also, well, what's important, and what I also already pointed out in the previous webinar, I feel like a broken record, but you also always should take into account the context in which you observe certain responses, behavioral or physiological responses, for a better interpretation of your results. So with these conclusions and / recommendations, I would like to conclude my presentation. And I hope there are some questions or comments in the Q&A. So thank you very much for listening. Kai.
Yes, all right. So now it's time to answer your questions. So thanks to you all for attending our presentation here. The questions will be answered here also live, and some of them we might be able to type in between. So I will go to one of the questions that the first one I think I can answer right away. There is a question of whether the slides will be shared. You will receive tomorrow an email, because you attended this presentation, that includes the recording of this webinar, but it does not include the slides themselves. In case you would like to have these slides, then it's best to contact us directly. So either me or Patrick, and then we can provide them for you. So that for me is with, you can see my name So that's k@noldus.nl, and for Patrick, it's patrick@noldus.nl. So you can contact us directly via that. All right. Let's see what else was asked, Patrick. There is another question here that goes a bit more into the specifics, and it is about how rapidly do two individuals interpret each other's actions and reactions. So that refers a bit to somewhere in the beginning of your presentation. Could you elaborate on that?
Let me see. Two individuals. For example, the example that this person uses child and an adult. Well, I guess that's quite quick. We respond to kind of what happens, the information that that reaches our brain through our senses. So if you talk to someone, immediately when you first meet someone, either a familiar or an unfamiliar person, your brain immediately draws conclusions about, it makes all kinds of assumptions about this other person based on how the person looks, the clothes he or she's wearing, the facial expressions, body language. So I would say you make an instant kind of assessment of the other person's kind of emotional state or bodily state. But I think we all know that we draw conclusions about someone else kind of instantly. But I think it's good to realize these are assumptions based on past experience, your experience with people from that kind of look the same or have the same profession. But I think it's important to realize that these are assumptions that our brain makes, are system one. So it's subconscious, it's very fast, but of course you could be wrong about this other person. You make assumptions, you think, oh, this must be a nice person because he or she looks in a certain way. But I think the proof is in actually communicating with this person. So to answer this question, how rapid is the kind of assessment of another person? It's instant, it's fast, it's system one in your brain. And when system two kicks in, then I think you come to a more reliable assessment of the other person.
All right, I think that does cover this question, Patrick. Thanks. I think indeed for more details, it's always possible to contact us, so we can dive a bit further into this question specifically. So let's move on further. There is also the next question anonymously put into the Q&A. What is the evidence for drugs like sedatives in influencing the facial EMG recordings? So, if one of us would be drugged, then so will we see some influence on the recordings itself?
Well, I would think, well, I cannot speak from my own experience. Maybe you can, Kai, but I can.
Neither can I.
No, but I can imagine if you take drugs, it could be sedatives or I don't know, things like LSD, things that kind of alter the state of your brain in any way. I'm sure it will have an effect on the EMG. So if you take a drug that kind of sedates you, it is likely, I think, that your face will be you will show less or fewer facial expressions because your body goes into a state of sedation or rest or you're completely drugged, so it's likely that also your face will be less expressive. The question also reminds me, we did a project where an artist and she was also a cosmetic kind of doctor. She used Botox, the substance that kind of removes wrinkles and from your face, but also kind of paralyzes specific muscles in your face. She used this for a kind of an art project to see how this substance change the, your facial expressions related to specific emotional states. So what she basically did, I think she used 20 injections of Botox, and then she tried to display anger or disgust on her face. But because of the Botox, her muscles were paralyzed, and she was not able to convey the facial expression that kind of represented her mental state. So when she presented kind of other people with her facial expressions, and she asked them to kind of assess what she was trying to express, they completely misguessed because she was not able to express her emotions through her face because of because of this drug. So that's a kind of more, I guess, more extreme example of how you can change your facial expressions through in an artificial way. And also interestingly, she also reported, because her face was kind of, she felt her face was a mask, and she said that she also felt less emotions. Her emotions were less intense. So I think this is a kind of evidence that not being able to kind of show facial expressions also has an effect on how you kind of experience these emotional states.
Okay, I think that's it also provides a more elaborate answer on the question. So indeed, there is clearly an influence, and what that exactly is, there's different ways that you can see that, as Patrick also explained. If you also would like to know a bit more about that, we do have a blog on the case that Patrick just mentioned on our website. We can also send it to you if you like, and just ask us via email. For convenience, I put the email also up front here, so you can write it down. That also reminds me that I will open a poll meanwhile while we keep answering the questions. If you also want to be contacted by one of our consultants to further discuss your specific research into more depth. So I will keep that open in the background meanwhile. All right. Whilst the poll is active, we will keep answering the questions you have. So let's see. Then there is this question about face reader and EDA. Patrick, so it's also here, would it be more easy to interpret the specificity of the emotion if you, for example, use face reader instead of an EDA, or is face reader also not very specific? What can you tell us about that?
Well, that's an interesting one, because I think well, I guess the kind of the reason behind the question is that if EDA is very non-specific, if you see a kind of a rise in EDA and you combine that with the output of face reader, would you be able to say, okay, I see an angry response that's also kind of represented by a physiological reaction in EDA? I think combining the two makes sense. So you get more specificity potentially by combining face reader and EDA. But at the same time, also with facial expressions, people vary a lot in how they express anger or surprise or happiness. But I would think that combining face reader and EDA, they could supplement each other. So I think it could potentially benefit the interpretation of the specificity of the emotion, as Cloelia, sorry, I'm not wearing my reading glasses, but yeah, it could. I think she's right on that.
Okay.
Yeah.
And I think it's time to also move on further.
Okay.
So then, as we can see from the next one, there is, like, if there is a technology of this kind that is possible to conduct remotely behavioral research, I think we can look at it from various angles. But let's see, Patrick, like, I think we also have to kind of give a definition of what is meant by remotely. And then we can either assume, like, okay, do we mean we want to have it in a non-invasive way, or really from another location?
I've interpreted it, but maybe it's because of Corona. It's kind of remotely testing participants who are seated in their own home, sit in front of a computer. So if that's the case, then yes, that can be done. For example, we have Face Reader Online, where you can send people link, and they use the webcam. The webcam is then used to analyze their facial expressions from a distance, so remotely. And also, heart rate can be measured. That's maybe good to mention, based on our PPG. So PPG is with the finger sensor that I showed in the presentation, but Face Reader is also able. I'm not sure if Face Reader Online is able to do that, but the standalone version of Face Reader is able to, based on the change in coloration of the forehead of the participant in front of a camera, Face Reader is able to quantify or measure heart rate. So that's really non-invasive and remote. So yeah, it is possible to conduct studies or do behavioral research remotely, using Face Reader Online, to kind of measure the facial expressions, to stimuli people see on the screen.
I think we can even add an example in which we also looked at devices that are used remotely by the participant. Like if you even want to add more precision, that you can send measurement devices to your participants who are remotely, and then also later integrate that data that you retrieve from there. So there are a lot of options, and it depends a bit on the specific context, like what are you exactly looking for. So I hope that so far it helps to get a general idea, the explanation from Patrick and me also about the options. But for the details, then please get in touch with us, then we can talk about that more specifically. Okay, so thank you for the presentation. That's always nice to hear. So, from Mercedes. And then it's the question is the discrete model of affective motion, relative to the circumflex model, was the pitfall of emotion studies in terms of inconsistency of psychophysiological responses. Okay, that's also interesting. It goes a bit more into the theory, like.
I think we have to take this offline, because I'm not sure what is meant by this question. So if this person can send me an email, we can discuss this, or we have a remote session on this, because I'm not sure if I completely understand the question.
Okay, then Patrick, I will put your email into the chat, so then Mercedes can send you directly this email, which is patrick@noldus.nl. So there we go. So Mercedes, for this question, we will have to go to the next one, so we can go dive more specifically into this question itself. What is exactly meant by this question? Although there was another question also.
Same person. Yeah.
Same, indeed. For a stimulus log study, which component of EDA is best to be analyzed, and why? Also, of course, good to know.
Okay. So for if you want to do a stimulus, kind of what's called a stimulus related or event related analysis of EDA, you typically use the phasic response. So what you do is if you remember, we had the kind of the slow graph, tonic graph, and then you see kind of instant peaks, which are called phasic peaks. In the algorithm, if you want to look at whether the peak occurred within a certain time frame after a stimulus that you presented, so typically it takes 2-3 seconds for a phasic response to occur. So what you do is you kind of normalize the graph. You kind of then extract all the phasic responses from the raw data, from the raw EDA graph. And then you can relate these kind of phasic peaks to the stimuli. So, for example, the BioPAC software, Acknowledge, you can specify where the stimulus was, you analyze the peaks, and then the software will tell you whether the phasic peak was related to a stimulus that was presented in the interval 4 seconds before that. So the phasic response is the one you use to do a stimulus-related EDA analysis.
Okay.
Okay.
So we also have the cover. Then we can move on to the next question. I see already some people saying their thanks. So you're very welcome. Then we go back to the sedatives question in the clinical situation. And when a patient is then sedated, how will that change the EMG expression and thus the interpretation of what the patient is experiencing? And if there's any studies on that. I think we already went a bit more into this question, like for example, with the part of Botox, that's quite similar. So I don't know if Patrick, you want to add anything on top of that from a more medical clinical perspective?
Yeah, well, I'm not a medic, but I can imagine that if you kind of when you sedate a patient, then this patient can still, I guess, experience anger if you kind of expose her to certain stimuli, but maybe then the facial expressions will be kind of prevented from you prevent this patient from expressing it facially. So I think then the interpretation based on the facial expressions, you would conclude, okay, this patient doesn't experience any emotions because we don't see anything on the face. But I think we've seen and studies have shown that, you know, what someone experiences is not always visible on the face. A person could be very introvert, or a patient could be kind of sedated. So it's a very interesting question, a relevant one. So I don't know of any studies that specifically looked at that. We can look in our database later, so I might come back to this and put it in the Q&A document that we will distribute.
Right. So then there will be more on that later.
Mm-hmm.
Then these two questions, they are put together from Fabrice, or Fabrice. Always have to check the right pronunciation. Which of the indicators would be more reliable for detecting acute stress, HR, ADA, or HRV?
Well, I would say a combination of the three, because HRV, typically you want to measure for a longer period of time, because otherwise you get the heart rate variability is influenced by kind of on the short term by breathing movements. But generally, acute stress or a stressful situation is usually measured using HRV, because depending on the person, heart rate can go up. If someone is really kind of a stressy person, so easily stressed, heart rate will go up immediately. Other people might be more calm, behaviorally, but also physiologically. So heart rate will not go up to the same level as the kind of more stressy person, but heart rate variability will definitely be decreased in both persons. So I think heart rate variability would be my first choice to measure stress or physical stress or mental workload.
Okay, so it indeed comes down to the HRV, but it's very important, of course, to have other measurements as well, so that you can compare and have a more rich picture.
Okay.
Okay, so now this goes into a bit more specific part. A pediatric dentist who also looks into facial expressions of children, which are very important during specific procedures. Now, this has to do with the lighting condition, so more about the application of the technology. Yes, so if there is like too much light, can this visual light be blocked, or is it possible to filter it out to make this facial expression more visible? How would you deal with that?
Well, that's a good one, because as the person probably rightly assumes, the light on the face should be kind of as even as possible for good reading of the facial expressions. But obviously, a dentist wants the mouth to be kind of lit more than the rest of the face. So, and that might kind of negatively affect the quality of reading the facial expressions. To be honest, at this point, I don't know how to kind of technically solve this without turning off the headlight. Let me also check with some colleagues. So maybe some of my colleagues, our colleagues, might have some experience with this, with people who are using kind of facial expression analysis in the same type of context. So I don't have an answer right now, I'm afraid.
I think in a more general way, it's kind of interesting to see that this is the opposite of what we usually are asked, because generally it goes about low light conditions and optimizing, of course, the analysis for that. But indeed, like having too much light can be a problem as well, and you have to be able to compensate for that. Now, best is always that we, with specific videos, we check it. But I see there's already an email, so we can dive into this more specifically with the person in question.
Yeah. So the email is in the chat, so that's good.
Exactly. Yeah. So thank you. Now, I think this is the next one is a bit of a shorter question about micro expressions or macro expressions. I think that you can tell with that a bit more about action units, probably.
Yep, I think that's a person I know. Hi, Efrain. Yeah, so there are basically two ways to detect micro expressions. So Kai already mentioned action units. So besides the kind of basic facial expressions like happy, sad, surprised, angry, face reader also is able to measure the activity of specific muscles or muscle groups, like the corrugator, like action unit, I think it's 4. So it also looks at the activity of specific muscles or muscle groups, and it gives you an intensity level. So, so you're able to kind of look at the activity, very deep activity, between a value of 0, no activity, and 1. So maximum activity of, I think, about 20 action units in the face. So this allows you to kind of look at what you could call micro expressions or activity of specific muscle groups. And also the basic emotions are also expressed on a numerical scale between 0 and 1. So even if a facial, one of the basic facial expressions is not very active or the intensity is not very high, you will get a continuous value for each of the basic emotions. So these numerical values and the action units allow you to also look at, detect micro expressions and analyze these.
Okay, I think okay, so we have the questions from the Q&A. There's one last question that I also would like to put forward here.
I have a couple of more minutes, Kai. I have another meeting at 5:15.
You have? Oh, you have planned this one for a bit later than this, but all right. I mean, you talked about so emotion and psychophysiology about that, and you also talked about how important it is that you have different input values. Now, that also implies that you have to be able to combine these together. And how do you actually combine these different data streams?
Okay, yeah, we as a company have an observation tool called the Observer, which was developed originally as a manual event recorder, but it has evolved into being a data integration platform. So you're able to not only manually annotate certain things that you see during a test, but also from the Observer during a live test, start physiological measurements, or start face reader, or start eye tracking using a special protocol that we have, communication protocol. So, and then after the test is finished, some of the data will be imported automatically on the same timeline. So in the example of face reader, the observer and BioPack acknowledge the physiological data acquisition system, it means that you immediately have your face reader data, physiological data integrated on the same timeline. So this means that you can, for example, look at when the level of happiness increased, what was the corresponding change in heart rate or skin conductance level. So the observer is our current tool that is able to kind of integrate and synchronize different data streams. And we are currently working on another platform, Noldus Hub, that does exactly the same thing, but in a more, I guess, more intuitive and more user-friendly way. So we have our platforms that are able to integrate different modalities for a better, kind of more insight into the response of your participants.
All right, so that does mean we have possibilities available to help with figuring out these different data streams and how to combine them and how to draw conclusions by comparing these different streams. I see that the answers from the chat, they have been answered so far. So thank you all for participating in our polls. Also, thank you very much for your questions. That's always nice and surprising for us to see, what comes up and, what new stories can we tell. If there are any other questions that you might think of later, again, please just send us an email. We will be happy to answer them. And then for now, I'd like to thank you all very much for participating and wish you a very good evening. Thank you so much.
Thank you all. Thanks for hosting, Kai.
Right, bye.
Bye-bye.